the intensity, the intellect, and the spirit of cooperation and teamwork that have come together in this room in the last 12 days or so is nothing short of remarkable. You have exceeded all of the expectations I had for this enterprise. It's been a huge team effort, as you know, and we all owe a very special vote of thanks to Gail for standing Arnold, who is unfortunately not with us, but she will get access to the video, which is being shot out of that camera on the tripod there. So let me not take any more of your time. I'm going to introduce your lead-off speaker, Jeremy Tang, who's an MD PhD student in the Rutgers Princeton program, the first year of PhD, and working in the lab of Sridhar Ganesan, who you recall from Monday. And Jeremy and all of you are going to bring this boot camp to a glorious close. There'll be a few words at the end about us getting feedback from you, and then there will be a networking session, which includes dinner, soft drinks, etc., and hopefully lots of lively conversation and photography, maybe even some reminiscences. So, Jeremy, over to you.
Dr. Barley. It was a great experience to be here, and we appreciate everyone who's here. And
Go on, you're very close to the mouth.
Like this?
Yes.
And we just want to thank everyone for supporting us and really giving us guidance through the entire process. So let's get started. Light-emitting machines, characterization of green fluorescent protein and its homologs. So we want to start our story off really with the Nobel Prize in 2008. 2008, in chemistry, as you all know, these three people, Shimomaru, Shelby, and Xian, were offered the Nobel Prize in chemistry for the discovery of GFP protein. However, it's not the GFP protein itself that really lended the Nobel Prize to them. Its application happened about 20 years. So, for instance, we used fluorescent proteins such as GFP, M-cherry, the citrine.
Sorry, just have this one.
Much better?
Yeah.
What things we have to do? Okay.
Do before. We had to make estimations and basically inferences of how proteins interact with one another, how they interact with their environment and whatnot. And once we were able to use fluorescent proteins to visualize these proteins as tags, we can really see these interactions, and they really opened up the doors for the rest of science. So today we use them in cancer biology, where we're trying to understand how does one malfolded protein really affect the rest of the system. In neurobiology, we're trying to figure out how synapses are working between neurons. And there's a whole bunch of other fields that we can really dive into. So for this presentation, what we're really doing is we're looking at these 5 different proteins: GFP, M-cherry, M-orange, M-citrine, and M-cerulean. And what we wanted to do was really characterize and understand how these structures and functions relate to one another and how they can be used in the future for other experiments or whatnot. So just an overview of what's going on today. What we did over the last 2 weeks was we isolated and purified the protein. Then we found its sequence and structure through DNA sequencing. We confirmed the identity of the protein through mass spectrometry and used a bunch of other tests to really characterize the protein, including fluorescence, circular dichromosome spectroscopy, differential scanning and calorimetry, X-ray crystallography, Nmr Spectroscopy. And then at the end, we're going to talk about what the implications of knowing this structure and function really will do for the field of science. So for the isolation and purification of proteins, I'm going to introduce our next 2 speakers, Ashley Gallagher and Ashley Grandin.
Does it work? Okay. Hi. Okay, so isolation and purification of fluorescent protein. So before we can do an analysis of a protein, we need to generate a large enough sample and harvest it. So we're just going to go over some of the wet lab procedures that allow us to do this. Okay, so this was done by transforming E. coli bacteria, and by doing this, we inserted a plasmid containing our fluorescent protein, and it is activated by a lac operon. And in order to make sure that we're producing bacteria that contains only our fluorescent protein with the plasmid is fortified with a kanamycin resistant gene. So when it's grown in an environment that includes kanamycin, only the bacteria that contains our plasmid is shown. So we got a nice picture of that.
So in the wet lab, we went ahead and purified the plasmid, but the sequence also had some tags that we put into it. So one of the tags was the his tag, and the his tag can be found on the amino terminus of the sequence. So in order to purify the protein, his tag is actually it binds really well to nickel. So we used a metal affinity column to go ahead and purify the protein. As the protein flows through the column, the his residues go ahead and bind to the nickel, and the rest of the protein falls through. So we did that with each of our washes, and as we continue to wash, the buffer removed the weakly bound proteins, and since the protein, the fluorescent protein has more of an affinity for the elution buffer, it fell through, and the his tag was removed.
Okay, so here is the result, a resulting electro, sorry, can you hold this? I don't know. Okay. So, yes. This is the result of gel electrophoresis, so we're probably going to go in depth with this a bit later. But here is just the result of putting in your subsequent washes into the gel. So here you have like wash 1, wash 2, and finally the eluted buffer wash at the end, which just contains the protein. And by putting them through the gel, the proteins are separated by mass. So, what? Oh, okay. So at the end, we just have, sorry, sorry. So this is just a thick band containing our fluorescent protein at the end, purified.
So we just also wanted to point out that the API tag and the his tag are not actually part of the sequence strand in their native state. It's something synthetic, something that we engineered and put into the sequence. The his tag helps in the process of purification, and the API tag is something that is used in biotinylation, which is something we didn't actually use, but it was something we didn't actually end up using, but it is useful for other purposes like image suppression.
It's black out of the clicker.
Yes.
Precipitation, sorry.
So, clicker. If you, sorry to stop you here, but if you can keep this closed, then this will work. Otherwise.
Okay, sure. I'm gonna go ahead and talk to us about the programming that we used.
Okay. Have is the nucleotide sequence of our proteins, or for the genes that encode them. So this really gives us a lot of information about the proteins that we are going to be learning about more today. So the first thing that we wanted to do with this was we wanted to put it into this program called BenchLink, which is going to do a couple things for us. The first thing is we need to translate this sequence from nucleotides into amino acids. So the program basically is just a whole bunch of different organizational tools that help you look at your nucleotide sequence. And now that we have an amino acid sequence, so you can see here, these are the nucleotides, and this is the amino acids in color below. The program does a couple other things for us. It tells us what the open reading frame is automatically. So that basically, for our purposes, is where the in between the start codon and the stop codon, which is not shown on this slide. And another thing that's really useful, as Ash was talking about before, is the AB tag and the his tag is automatically located using this program. So it's right at the front of the sequence right here, and the his tag is right here. And this is just something that we want to point out with an annotation so we can keep it in mind as we are analyzing our sequences. So now we have the translation, and I really wanted to include this as a slide because this is really the fundamental data that we have up to this point. But really, as you can see, this is not very easily interpretable, so we need to do something more with it before we can actually get any useful results out of this. So the first thing that we wanted to do now that we had our translation was we wanted to look in the database. And in the database, what we're trying to do is two things. One, we want to make sure that the sequence that we have is consistent with the sequences that are already in the database for this protein. And we want to find other sequences which are homologous for different proteins and see exactly what the percent identity is. So we did a BLAST, which is a program that is going to look for homology between sequences. And what we found was that for each of the protein sequences that we had, just as expected, the most identical one was itself. So that's good. And then from there, we could see some of the homologous sequences. So for this example, this is a BLAST that was run on M orange. You see that it's kind of hard to see, I apologize. M orange is the first most identical sequence that was found. And then you could see M cherry, and you could see a lot of red fluorescent proteins are also very homologous. So this is kind of a clue at this point that there might be something going on here. There's some sort of similarity between the sequence for M orange and M cherry. So now what we wanted to do is take these amino acid sequences and align them. The way that we did this was we put it into a program called JLQ. And JLQ does a bunch of things. They're all listed here. It aligns sequences, makes phylogenetic trees, and allows you to compare structures as well. So what I have here in this image is a multiple sequence alignment of the 5 different proteins. Basically what I wanted you to get out of this picture is that you can see where the amino acids line up very well, and then where we've had to insert gaps to account for indels in the sequences. So basically this is done using an algorithm. The program that does this is Clustal Omega, Clustal X. There's a whole bunch of different ones out there. The one that was done with this is Clustal Omega. And what it does is it's going to use some sort of algorithm that inserts gaps where it needs to be in the sequences. And this is really important because this is our hypothesis for which amino acids are homologous with each other in the sequence. And it's either going to make or break our phylogenetic tree that we really hope to get out of this in the end. So here's an overview of the alignment. Each of these bars is one of the proteins. The top two are cherry and orange, and then you have GFP, citrine, and cerulean. So each of the colors is representative of a different amino acid. You can see where they all line up very well is where all the amino acids are identical. A couple things that I wanted to put out on this is one way of seeing the consensus between the different amino acid sites is this yellow bar at the bottom. The height of the bar indicates the consensus between them, so the higher the bar, the more aligned they are. And then also these white spots are gaps. One thing that I think really should be easy to notice here is that there's sort of a pattern emerging between them. You could definitely tell by looking at this that the top two seem to have a pretty high degree of consensus with one another, and the bottom three have a high degree of consensus with one another. So this is another clue that we have that there's something going on where cherry and orange are kind of grouping together, and the other three are grouping together as well. So here's our tree. The tree also comes from the same program, JLView, which uses another algorithm to figure out what this tree would look like. And it's pretty obvious what you can get out of this. Cherry and orange are grouped together down here in this taxa, and citrine, GFP, and cerulean are grouped here. I think that this is something that is really important, and it's something that should be kept in mind throughout the rest of this entire presentation, because this bifurcation is probably something that you'll see quite often as a pattern in all the other sorts of data analysis that will sequences. You could have a prediction of secondary structure through looking at the amino acid sequence. So we'll talk a lot more about these sorts of structures in different data analysis methods like X-ray crystallography. But one thing that I think you can get out of this, at least, is it shows you where the green bars are for beta chains, or what would be predicted to be beta chains, and the red bars are alpha helices. And this black bar indicates the quality of the prediction. Is it very likely or not? So really what we're seeing is there's a lot of beta chains here, with a few alpha helices, and keep this in mind as we look at CD and different things later on. Two more things. This is a chart which shows the different masses as predicted by the amino acid sequence for trypsin fragments. This is if you had the protein being digested by trypsin, where it would be fragmented. These are the different fragments of the amino acid sequence, and this is the mass in Daltons. The reason that we would want to know this is because it's another thing that we can look at with mass spectrometry. So we could use these results to compare with mass spectrometry. And finally, another thing that's very relevant to mass spectrometry is just the mass of the whole chain. So we can figure out what the mass of the chain is by the characteristic mass of each amino acid, add them all together for each sequence, and we get this prediction. And I believe later on we will see these numbers again when we look at mass spectrometry. So moving into that, we will have Jackie Wong, who's going to talk about mass spectrometry now.
All right, so mass spectrometry. Before we move on to mass spectrometry, I think it'd be best to reiterate some of the points highlighted in the slides about the protein purification column and the electrophoresis. So basically, eggs washes, right? And as you can see, our proteins are completely pure. There are contaminants or perhaps fragments of the protein. So that brings the question of like what exactly is in our solution, the final test tube solution that we hope is mostly purely the protein that we want. And so for that, we turn to mass spectrometry to characterize what is in our test tube. And so electrophoresis of a protein is not quite clear. It's not a clear characterization of a protein, but it's a quick and easy method to check if our protein purification method was good enough to be like experiments such as NMR or CD. And the larger bands in our gel electrophoresis can indicate that some amount of purity is again in our supposedly pure sample. So we need methods to confirm the right species of protein from the database that we work off of. So with that, I will turn the mic over to Marini, who will go into further detail about mass spectrometry.
What's Marini's last name?
The particles then get deflected, and then the particles get detected. So the 4 steps are outlined here, and the different rates of detection give rise to different molecular weights as observed right here. So what you're going to see right here are characteristics of a mass spec peak, and depending on the intensity, depending on how well it's detected, you'll get larger characteristic peaks. And these peaks, if you zoom in on any of these sections here, you're going to end up with something like this, where you're going to see a mass to charge ratio here and a relative abundance or intensity right here. And this mass to charge ratio, if you want to find the molecular weight of your, whatever you're studying right here in species, you're going to just multiply this charge by this mass to charge ratio, and you're essentially just doing m over z times z, and you're going to get your molecular weight. And if you zoom in even further, you can actually match these, like sequence Cassandra mentioned, you can actually match these to certain sequences right here, and you can see how you can get a prediction of what exactly your molecular weights could be. And you can see if your mass spectrum is correct, and there's also an error provided. Now, we used MALDI-TOF to analyze our proteins. So what happened was, it's a laser detection system, and what happened was, you can see 3 characteristic similar weights right here with the, as we mentioned before, the 3 Cnidarian derived proteins, and you can see the 2 proteins that are derived from coral also being characteristically different. You can see that they're around 31.2-ish K Daltons, while these are around 31.3 to 31.45-ish Daltons. And you can see that these three are very characteristically similar, while these two are characteristically similar. And the last thing we'd like to go over with mass spectrometry is that when we looked at the sequence of the protein we were analyzing, GFP, we could see that this unique region right here, the chromophore, followed by the residues following the chromophore, you can see that this was the one section that this database could not actually identify because it did not detect it when we were performing the multi-top analysis of our protein. And every other region was identified, so this is also the key difference between all the proteins because each has a unique chromophore. And with that, I'd like to pass the mic off to Stefjord.
Hi everyone. So far for our protein, we have a very good idea of the sequence from sequence analysis. We have some kind of affirmation and confirmation from the mass spectroscopy. And we have an idea. We made some kind of a prediction using our sequence data to predict the secondary structure. And one way to actually analyze and go to lab and experimentally determine the secondary structure is circular dichroism. Next. So very briefly, I'm going to say why we do this. So circular dichroism is a very useful tool for secondary structure prediction, among many, many other things. The basic principle is different secondary structures respond differently to circularly polarized light. We have left circularly polarized light, say that 10 times, and right circularly polarized light. And depending on if the structure is an alpha helix or beta strands or just random coil or triple helices, they're going to have a very different signature. And these are studied very well. For some reason, the reference didn't make it here, but by Norma Greenfield, which has an amazing review on this. So, among many other things that we can determine using circular dichroism, we can at least have an idea of the secondary structure of the protein. And after we do the experiment, we can see which one of the signatures we can mostly see in our protein, and then we can deconvolute that and determine the percentage of each one of the secondary structures. So I'm going to give the mic to Zahra, and she can explain our results.
Hi. So,
as we saw in the DNA sequencing, we predict that we have most beta sheet here because we expect that we have most beta sheet here. So here you see the raw electricity versus wavelength. So we have orange. This is supposed to be for the orange and cherry protein, which look like more or less the same. They are very close to each other. And we also have the other three proteins here. They group to each other. And as you see, to compare with this, we can see that this is more or less the same as the number 2, the beta sheet, and that's what we expected. So you see the minimum is more or less taken in 215, which is the same as what we have here for beta sheet. So Tahir, Yusuf Ali, and Daniel Quentin will talk about fluorescence.
Is this good? Okay, thank you, Zara. So I'm going to give quickly a brief overview of the way fluorescence spectroscopy works, and then Danielle's going to tell you the results that we have for our proteins. So the essential principle of fluorescence is that the structure of each molecule, each chromophore, is a combination of the electronic structure, or the configuration of the electrons, and the vibrational structure, or the structure of the nuclei. So now what you're seeing here is that when we shine a photon at a molecule, the big blue arrow and the red arrow are electronic transitions. That's when the electron gets excited to a higher level. And electrons can only have quantized energy levels. So it can't just go to a level that's right by it. It has to jump only to the next available level that it can get. But now on the other hand, when we shine light and excite the chromophore, there is a certain period, a fluorescence lifetime, in which because we had shined so much photonic energy on the molecule, not only did we excite the electronic structure, we also excited the vibrational structure of the nuclei inadvertently. So those little mini black lines you see there are vibrational levels with the electronic level fixed. So fixed electrons, but variable nuclei. So what you're seeing during this fluorescence lifetime is that the molecule has been excited electronically, but is relaxing the excess vibrational energy to its vibrational ground state. And then after that fluorescence lifetime, then it jumps back down to the electronic ground state. But because of that vibrational relaxation, some of the energy of the original absorbed photon has been dissipated away as heat. And that's why the emitted photon's wavelength is slightly different. So what we can gauge from this is basically two things to first order. The first is the electronic structure, particularly the gap between the first and the ground state and first excited electronic levels. And secondly, we can get a sense of the vibrational structure by looking at the nature of the vibrational subbands that relate to the difference to the fluorescence lifetime and the difference between the absorbed and emitted photons. And so that's the way it works. And so now Daniel's going to tell us what we got.
All right, so why do different proteins absorb different ranges of wavelengths? Well, we can answer this in terms of structure and chemistry. So each chromophore has a different structure, in particular their positions in nuclei and their electron orbital configurations. As a result, the excitation gap between the lower quantum energy level and the higher quantum energy level differ for each protein to protein. This is an excitation spectra that shows the maximum peaks for each of our proteins. So here you can note three things. One, that alright, so this is the absorption spectra and this is the emission spectra. If they absorb it at a particular wavelength, they emit it at a higher and longer wavelength and lower energy. Two, you can note that the peaks for orange and cherry are very similar, so we can use this as evidence that the two may be evolutionarily similar. And these are the respective peaks as well. These are our beautiful results of the fluorescence that we worked hard for. They're very pretty. And next, I'll pass it to Mike Olson, who will talk about differential scanning calorimetry.
Thank you. So I'm just going to talk a bit about differential scanning calorimetry and how we used it to help characterize our proteins. So differential scanning calorimetry, or DSC for short, Basically what this does is that it measures temperature change in a sample of interest relative to a sample whose heat capacity is already known. In this case, sample of interest would be one of the proteins, and the reference sample would be just the buffer or solution in which the protein's exchange of heat with the surroundings, which is important when you're trying to find this change in temperature. So from a plot of specific heat versus temperature, enthalpy of unfolding for each protein can be determined for that sample by finding the area under the curve just by integration. Plots can indicate stability and folding patterns for the fluorescent proteins, and that's useful for us because that's exactly why we would want to use DSC. We would want to see exactly how these might fold into the respective structures. So now this is the UV melting analysis. This was actually done before DSC, and what you're seeing in this plot is over here at this wavelength. You're seeing the UV light that was used to excite the different chromophores in the proteins, and what you're seeing along here is the different wavelengths of emission for these chromophores. One thing that's interesting about this is you'll notice, some of them have like 1 or 2 peaks, but especially with orange and cherry, you see peaks that are at these lower wavelengths. And so I'll get into that in a little bit, but that is important and shows something different about the behavior of these proteins and perhaps their structures. Okay, then this was also part of the UV melting analysis. What's being indicated here is pretty much where the UV the absorbance by UV pretty much just levels off, and which indicates that fluorescence has saturated, and indicates an irreversible change in structure. It's gotten to the point where the structure is just unfolded and does not look like does not have the same does not resemble how it originally did. One thing to point out here is that the cerulean, citrine, and green fluorescent protein all are at relatively lower temperatures when you see that occur, when you see that leveling off. Whereas orange and cherry are significantly higher temperatures when you notice that irreversible change in structure. Okay, so what you have seen here is the actual results from DSC, and this is just an overlay for all the different proteins. One thing I'd like to just point out real quick is that you have these 3 peaks for citrine, cerulean, and GFP, which are just one nice sharp peak. Whereas with orange and cherry, there's something that the spectrum is a little bit more involved. So I just want to go into a little more detail about cherry. What you see here is you do see one very clear peak around 97 to 98 degrees, but you see these 2 basically just lumps that are very close together around 65 to 80 degrees, and this indicates something interesting. It indicates that there's something more involved going on with the structure. And I also want to just point out real quick that you actually notice somewhat of a difference between the left side of the curve and the right side of the curve. That's because there was some protein aggregation that occurred as a result of the protein unfolding and the unfolded parts basically coalescing, which was something that's not ideal because it's harder to find out the temperature at which this transition is occurring. And just to make it clear, each peak that you see here corresponds to a transition temperature, the center of each peak. Okay. Now with the orange, this is where it really gets interesting because you can see 3 more clearly defined peaks, 2 of them being at lower temperatures and one of them being 97 to 98 degrees. And what I'd like to point out about this is with cherry and orange, you're seeing something distinct that's happening between them that's different from the others. And in particular, I could see 2 possible explanations for this. One of them being that these 2 peaks that you see correspond to smaller fragments of each of those proteins, which may be coming together in order to form the overall structure, which would correspond to this transition. Another possibility is just that there are smaller fragments which are melting at these or having a transition at these temperatures, and are just present in the sample basically as contaminants. And so both of those indicate though that there's something different about the cherry and orange samples that you don't see in green, citrine, or cerulean. And this summarizes the data in a table for each of these. And just to point out, the cherry and orange, you see similar temperatures for the transition, and similar delta H's for the unfolding, or enthalpy changes for the unfolding. Whereas citrine, GFP, and cerulean are noticeably lower. One thing to point out about this as well, though, is the reason why these seem like very high temperatures, very stable proteins, is that these were engineered in order to have extra folding capability. But you still see a similarity between these types of proteins that's important to consider when you look at how these proteins might have evolved over time.
Help. Pretty much covered. What is X-ray crystallography? So the method is based on physical property of light to bend when it encounters the obstacle. If the obstacle is ordered structure, so the waves bend in a certain pattern, and some waves happen to get into antiphase and cancel each other, and some waves going to facilitate each other. And as a result of diffraction, you will see the picture, which is kind of black dots on a film, or you can detect it with CCD camera. And here's how the diffraction picture looks like. So back to proteins. In order to proteins usually in a solution, they move randomly and they don't form any ordered structure. So we need to crystallize them. And our wet lab team successfully get the crystals from lysozyme protein, and that's how they look like. So we were able to pick up those pretty crystals with very fine, very fancy loop, and shoot them with X-rays. And we actually got a few pictures from different angles. This loop is rotating, so you kind of get a series of pictures. And by this pattern of diffraction, you can calculate what's the shape of units that form this crystal. So you can know the shape of these particles that form this crystal, and it's represented in an electron density map. So it's usually depicted as a mesh, and the mesh surrounds the space of electron density. So now we know the shape of the particles. How we can determine the coordinates of atoms? So we know the primary sequence, and we start, the crystallographers start finding the places for the big amino acids, like here so they trying to match the tryptophan, tyrosine, and the phenylalanine, and then kind of trying to feed the rest of the chain into the electron density map. And the results are amazing, because nowadays crystallography can get the protein structure with high resolution, and the overlap is pretty much well aligned. So the major complication of this method is to get these crystals. And this slide is just to show you how tough the life of crystallographers are. So there are many factors that affect crystallization. So the most obvious one is pH, ionic strength, and temperature. But you also have to consider the protein concentration, and it should be relatively high. Protein purity also should be high. So you can use some additives to get rid of water molecules, because they form crystal, and those crystals on acid, they produce high background on your diffraction picture. So you can use cofactors, substrates, and ligands of proteins to stabilize the structure. You also use a lot of reducing agents, because X-rays, they are produce free radicals, and they bad for your protein. They change the structure, they oxidize amino acids. The list goes on, and there's also some weird factors like vibrations, drop well volume, and all other random things. So this is our GFP protein. This is electron density map downloaded from PTB website. So that was kind of raw data of crystallographers. a little bit processed, and now we're trying to fit our GFP protein into this picture, and I'm gonna pass my microphone to Pauline.
Thank you. Here is an example of a chromophore of a fluorescent protein. This is the chromophore of the cerulean fluorescent protein, and I'm just going to show you a practical demonstration of what Ina was talking about using the that mesh you get from X-ray crystallography for to model actual chemical compounds. So here is that same chromophore from a slightly different angle with the X-ray crystallography mesh around it. This is pretty good resolution. This is probably about an angstrom resolution. As you can see, the mesh very closely follows individual atoms. These are the actual electron clouds of individual atoms, supposedly. You can get even very specific parts from the inside of the protein, such as this chromophore, and the chromophore is at the inside of this beta barrel. This one particularly is M cherry. You can't actually see the chromophore; it should be around here. Using X-ray crystallography, you can see that all the beta sheets form together into this basically the same beta barrel with an alpha helix inside. Here's an overlap of all of these beta barrels. This is all the fluorescent proteins: GFP, citrine, cerulean, cherry, and orange. The beta barrels all generally overlap. The main differences are the chromophore in here and a few little side chains hanging off to the sides. Here is a comparison of all of the chromophores. As you can see, most of the chromophore is basically derived from 3 fused amino acid residues. There's one conserved glycine in here. There is a small amino acid here, which depends on which protein you have, what amino acid that was derived from. You can see here is a methionine for cherry. Cerulean, GFP, and orange all have threonine, and citrine has a glycine much shorter. And for this residue, it's not actually a residue; it's derived from a residue. For this one, most of them, with the exception of cerulean, use a tyrosine. This one has a tryptophan. And here we have this conserved glycine is very important because we have a hydroxyl group off of that glycine. It's right here in this one. This interacts with a conserved arginine and glutamate, and the electronic interactions between these residues are very important for the fluorescence. Are you taking over here?
Alright guys, so using the PDB website database, we searched up a list of molecules that were similar to the fluorescent proteins that we used, and we picked the most dissimilar one. So what we have is nitrogen, and how we know that it's the most different, we used the RMSD value. This one apparently had one of 2.99, which according to Professor Burley is very, very different than ours. So the only thing that's the same with ours is you can see the beta barrel, or part of the beta barrel is similar to the GFP protein, but even then it has like a tail on the outside over here, and the middle part, the part that gives out the fluorescence, is completely different. And you can see the expanded view of that. And this is a culmination of our experiments, is the phylogenetic tree. All right. So according to the other experiments, I think it was in DNA sequencing, those slides, we had a prototype of this. So we start off with some historical protein, probably also green. But we have GFP as the main head of the family, so to speak. GFP gives rise to two different groups. This is supported by our data because orange and cherry are completely different than the enhanced GFP, the citrine, and the cerulean in terms of data collected and their structures. So as you can see, the citrine and the cerulean split off from the main branch of GFP, and orange and cherry are on their own. We put nitrogen on the side to show that it's completely different than these, but it also has some sort of beta barrel structure, so that's the reason we included that. And this is thanks to Professors Anavaparu and Kare. So I'll pass it up to Neha Sikka for the NMR.
Okay, so extra crystallography is great for finding our structure, but it has a lot of limitations. In addition to it being a horrible time to try to make a crystal, you can't learn everything you need to know about a protein from this static model. So one thing that we would like to do in the future to analyze our fluorescent proteins is to consider Nmr Spectroscopy, which stands for Nuclear Magnetic Resonance Spectroscopy. So this is better for fluid. It's a test that can analyze structure, dynamics, reaction state, chemical environment. And as a general idea of how it works, you load your protein into this machine, and your protein has one type of hydrogen. So in our case, we used ubiquitin, and ours just had normal hydrogen, like a proton. And then you put it in heavy water, which has deuterium. And in the machine, all the hydrogens exchange with all the deuterium. And by measuring the energy and time that it takes for these exchanges, we can make analyses about where these residues are. So for example, a higher residue would be more shielded by the protein structure and possibly hydrophobic. Since we would like to do this in the future, but to see what we would get, we went to look at some literature. And one of the pieces we found by Nua in 1996 actually performed NMR on GFP. So it's not only just the chromophore, but also that environment that's so important to study. This is the NMR that we experimented on with ubiquitin. It's actually in the Life Sciences building, so thank you for that. With your NMR, there are results you can get. This is an HSQC, which stands for Heteronuclear Single Quantum Correlation. We studied to learn about flexibility of your protein, or just general composition, which is a nice way to visualize the backbone. And that being said, I'd like to introduce the one and only Kenneth McGinnis to kick off our lecture.
Well, good afternoon. And just to let you guys know, we have a wonderful spread outside waiting for us, so I'm not going to hold you up for too long, okay? Actually, I spent a great deal of my undergraduate working for these guys and ladies, and it's been a wonderful experience, so it's very pleased to see that they're out there. So, moving forward, as all great events, when they come to an end, it's great to discuss and look back on what we learned and to improve as we would this whole procedure we've done in the last 2 weeks, and it's been a phenomenal experience working with each and every one of you, because we all have our own perspectives. In order to give in a timely and polite manner, you know, it's difficult to culminate everyone to do that. So, all right, so just the synopsis before we go forward. So if we were going to publish a paper, you know, we'd ask ourselves, what have we done? What do we have? And what we need to do. So we've looked at sequence. We made sure we had the plasmid that we wanted. Our friends gave us plasmids. They gave us some cells. They said, you know, go make some proteins. So we had the right plasmid. We made the proteins. We did mass spec. We found out that we had the right proteins. Next, we did a culmination of structure-function relationships by probing by different biophysical characteristics. So we have predictive models that predict what our structure should be, what our protein sequence should be, and from those predictions we're going to choose the experiment that will best suit our needs, as well as understand the limitations of those experiments. So we use CD. We found that it's primarily a beta structure. The GFP variants. We used X-ray to determine the 3D and tertiary structure of the each variant. And so the probe its function, because it's important, because as was previously discussed, is that there's a utility for these beasts, in nature as well as in our own labs. So we use fluorescence and we use calorimetry as probes to determine, and calorimetry in particular, because where we put these in one environment is important, because if they don't do what they need to do because they unfold, it's important to know that, because that's not useful anymore. So what has our data has shown us is that fluorescence, these variants, they differ in biophysical characteristics. And one of our hypotheses was that they've diverged evolutionarily. So I hope we've come to show you some data that would suggest that, and we're definitely open for suggestions. So what everyone has just shown you and told the story of is the basis of retrieval as the primary evidence of getting this ability to analyze the data. So we were given a task of 5 plasmids and cells. We were told, please make these fluorescent proteins. The method we used, we tricked some E. coli cells to making what we needed. And we characterized and we confirmed what we had. So how we did that, and this is the beauty of this program, because this is interdisciplinary. Each one of you speaks a different language. However, English, a Chinese dialect, or Indian, any dialect you speak, you all need to come to some basic understanding of each other to communicate in life. And that happens in science. But this language is made into different variations of science. And how we retrieved was through molecular biology and bioinformatics, as we showed. So, as with all great retrieval, there must come great characterization. So, what we did was we developed a hypothesis. These fluorescent proteins are somehow evolutionarily linked. So then we developed a model. So we built a phylogenetic tree, which we then went back to each and every experiment that we went to and said, does each experiment give us this data? Does it back up our phylogenetic tree? So then it was needed to determine the experiments that we were to use to test this phylogenetic tree model. So then we said, hey, what's going to happen? What are we going to get? What do we expect? Then we had to come to a point in which we said, did our expectations meet the outcomes of what we expected? Very important key. And again, this culmination of languages came together. And many of us weren't fluent, didn't speak it, haven't heard it before. However, in this boot camp, we were exposed. The exposure of this language can be built upon in future and used as a reserve for when we come across a problem that needs a particular experiment or a particular piece of information that we've come across. So we're not experts in what we've known. We just know a little bit of information. So spectrometry, calorimetry, spectroscopy, crystallography, fluorometry, and bioinformatics have encapsulated all of what we've done. So this is the beautiful part right now, right? Because we have this evolution, but we have this key ingredient of novelty. Because we have this extracted protein from nature, the jellyfish, which is green, right? But guess what? We changed it to fit our needs. And this was a very big theme of design. If we take what nature's given us and look at disease states or pollution in the environment and design weapons and tools and proteins that will take care of that for us. That's a powerful tool. But we've shown just a glimpse of that using these GFP variants. So what I want to put out there is this idea of divergent and convergent evolution. So what I show you here is a picture of GFP and the different variants diverging from one GFP molecule. And that is based on the engineering of GFP. Individuals have sat countless number of hours determining the correct amino acid to mutate in order to get a different variant. And they've done it in such a way that they do not overlap with each other, so that they can be used in cellular situations to determine where proteins are made, where proteins are degraded, where proteins are coming together and making, they're co-localizing, so you can determine whether or not a protein is relying upon another protein. That's a hell of a tool. Love it. So, and the story about Nitagen is beautiful, because this could be a story of convergent evolution. Is that, as this is many times has been told, is this nature as an idea of being parsimonious, meaning let's go to the ground energy state of a fold of a protein and the most stable fold that there is. Nature has come up with a model that folds into a beta barrel, but it doesn't fluoresce. Why is that? What organism, this is from the mouse organism, why doesn't the mouse need to fluoresce? The mouse doesn't live, 1,000 leagues under the sea, where it needs to create its own light and interact with other individuals who don't have light either. So that positing an idea of why the fluorescence, it was found in the jellyfish to begin with. So the idea of this being a homology relationship, and the idea of why did the jellyfish develop this ability as opposed to the mouse not. So each and every one of us, we told a story. And everyone has their own story to tell scientifically and in life. The heroes of this room are the scientists that spend countless number of hours passionate about what they do, and their willingness to talk and say what they've done, and give their time and effort to determining what makes the world tick. The tools that we've used to show a little glimpse of what life is like if we engineer it. 5 fluorescent protein variants. And the needs that we have, because we have these tools, but we need to understand them. We need to characterize them so we can control them, so we don't have any unknowns. So we can build upon those systems and say, I know what this protein does, but I want to add complexity to it. So I need to characterize it using all of these biophysical characterizations that we have. Understand their limitations, so we can design new experiments that will go forth and erase those limitations. So how have we done this all? We've retrieved, characterized, and we've synthesized. And now it's time to go utilize these tools in any which way we found useful. So as we see, we like protein control. So my deepest and most grateful acknowledgements at first, Dr. Stephen Burley. Thank you for your energy, and thank you for your willingness to take on this role and facilitating such an amazing series of events. Dr. Gail Arnold, which I don't know how many emails she sent to everyone in the back who arranged this, countless number of hours. Thank you for her. She will be on Skype in about, she should have been maybe, actually 15 minutes she'll be on Skype. So if you want to say hello, wish her well. She's going through a tough time with her family. Please stick around and say hello. The guest lecturers, you are an attribute to this society that is very valuable. You are great citizens. Thank you very much for your time, your effort, your thoughtfulness, your willingness to work through problems. Workshop leaders, thank you very, very much for your time as well. The knowledge that you've, it's greatly appreciated. Boot camp participants, everyone who's ever been in this room during these last 2 weeks, you are valuable. Because we can't do this alone. We don't work in a box. We need communication, we need social events, we need to express our ideas no matter how crazy they are, no matter if I can't speak what they are, I can't articulate it, because sooner or later if I talk long enough, you'll understand. And of course, we wouldn't be here, no one would be here without Rutgers. So, thanks.
So may I say a few more words, please? So thank you one and all. That was great fun. I think I speak for all members of the faculty and the technical staff at the university who participated in the program in saying that you've done an amazing job. And you have, as I said at the outset, you've exceeded my expectations. You got it, and getting it involved two things: getting the science, putting together this complicated jigsaw puzzle, and getting it in the sense that you can only do that through teamwork that involves people with different perspectives, different skills, different analytical bents, different ways of thinking. And it's bringing all that together that makes modern biology so exciting and so successful. And success can come in many, many realms. Some of you are MD PhD students. You're going to go on. You're going to do medical. You're going to take care of patients. You're going to do medical research. You're going to influence the lives of patients and their families. Contribute to health and wellness. Similarly, those of you who are involved in engineering, in the basic biological sciences, and some of the quantitative sciences that can be brought to bear in biology can contribute not only in medicine, but in our basic understanding of life, and also the interactions that occur on this planet that are so vital for our sustainability as a population now of many billions. We have to do much better at taking care of the environment. A knowledge of biology and a knowledge of how to engineer proteins, how to modulate things in a careful and thoughtful and responsible way can make a difference going forward. We're going to have to, in our lifetimes, we're going to have to adjust to significantly higher sea levels, significantly higher temperatures, threats to species, etc., threats to our safety and security. And much of what the biological sciences have to offer will contribute to success in that endeavor. I again say that Gail Arnold was the person who made this work, and I'm very, very sorry that she has other responsibilities which have appropriately pulled her away today, because it would have been great fun for all of us to reiterate our thanks and our admiration. She worked sort of 80 hours a week for months to bring this off, because she had a day job that didn't include putting this whole thing together, but here we are, it worked. When this was announced, it was announced as the first annual, if you recall, and that was done that was perhaps gutsy, but it was done with some confidence and also a sense that this was, a belief that this was a way to make a difference here at the university. And we absolutely are going to do this again, but this was a proof of concept experiment in many ways. It came together very rapidly. Not all of it was perfect. Some things worked very well, some things didn't work quite as well, and we want your feedback. Just this afternoon, Kenneth, thank you, posted on the Sakai site a survey, and we need each and every one of you who participated either as instructors or as students of all ages to complete that survey. You can do it and indicate your name, you can do it anonymously. The important thing for us is to get the feedback. There's a whole series of quite pointed questions there, but the very last section of the survey is free form. Just say anything you want to say there. You can email it back to Gail. You can handwrite it and slip it under the door in the dark of night so no one knows you did it. Whatever works for you. But we absolutely need your feedback because we want this opportunity in 2015 to be even richer for the people who come after you. We do plan in 2015 to repeat the GFP theme, having had such a successful run with those 5 proteins. In 2016, 2017, we'll probably shift gears and we'll probably look at another protein family that offers similar opportunities. So I hope some of you will be back when we change gears. I think it will be helpful for all of us next time around if we could put more emphasis on macromolecular interactions. You saw about surface plasmon resonance, you saw the power there, but there are other tools that can be used to study interactions among macromolecules that are vital for control of cells, metabolism, etc. And we're going to build that in. I saw that as a deficiency, but given where we were, that was, in terms of the planning, that was a deficiency that we simply had to accept. So any and all feedback is good. I hated it, and the reasons why I hated it is equally valuable, perhaps more valuable than I loved it. I loved everything about it. There's a good spread outside, I'm told, and I hope all of you have a great long weekend. Come back to school on Tuesday morning, energized, ready to go. Thank you for taking 2 weeks out of your holiday when you could have been skiing or earning money or doing other things to do this. One thing we'd like to know is whether you would do this for credit, whether or not you do this on a pass-fail basis. All these questions are asked in the survey. Please answer them as forthrightly as you can, and help us make the opportunity even richer for next year and for subsequent years. I want to just conclude before we bring Gail onto Skype and start with the food and the networking, as it's called. You can't have a party at Rutgers, I'm told, but you can do networking. So we're doing networking. I want to acknowledge the efforts of Helen Berman and her colleagues before I arrived at the university in making the case to build this beautiful building and to create this Center for Integrated Proteomics Research. The notion here was to bring basic sciences together with basic biology, together with the quantitative sciences, with medicine. Those 3 things to build a research environment that would enhance Rutgers, and the symbol of that, those 3 things, of course, is the collagen triple helix that's outside that you've seen every day when you've been coming in here. That was one of Helen's major contributions, and a beautiful piece of art to symbolize what the building's all about. So thank you again. I take my hat off to you guys that did a spectacular job.